Thank you very much.
I'm Ethel Blessy, and my presentation is entitled Discovery and Development of New Chemical Compounds with Antifungal Activity from Marine Endophytic Fungal Sources. I'm being supervised by the following people. Okay. So, antifungal agents eliminate fungi pathogen from the host with minimal toxicity to the host. However, such antifungal agents are very difficult to develop because fungi are eukaryotes, and so we have genetic similarities between fungi and human beings. Certain drug targets found in yeast, for instance, are also found in humans. So the ideal antifungal agent should be able to differentially target the fungi pathogen. And the limited number of antifungal agents being introduced and the misuse of the ones that we have available now have led to an increase in antifungal resistance, and this is a major source of concern. There's also a rise in population of immune-compromised people who are prone to chronic fungal infections, such as people living with HIV and AIDS, transplant patients, and infants. The current antifungal agents used in clinics include fluconazole and amphotericin B. And recent reports have shown the emergence of resistant fungal species. Therefore, there's an urgent need for us to discover and also develop new antifungal agents. Terrestrial endophytic fungi have been identified as an excellent source of bioactive metabolites, and some of these metabolites have even been developed and are being used in treatment of patients in clinic. However, marine endophytic fungi have not been explored as compared to their terrestrial counterparts when it comes to the treatment of human diseases, but they are also very good sources of bioactive metabolites. These are just examples of some of the isolates we have in our lab. So Felicio and colleagues isolated endophytic fungi from marine red algae, and after going through the fermentation and product isolation process, they arrive at cytocalycin D as their bioactive metabolite. And then this cytocalycin D exhibited a very strong antifungal activity against Cladosporium species, which shows that it is possible for us to also isolate other compounds from endophytic fungi, which will be very effective against other pathogenic fungi. Therefore, the aim of my project is to discover and develop potential antifungal compounds from marine endophytic fungi using yeast genomics guided approach. And for the objectives, I'll be isolating secondary metabolites from marine endophytes and then determining their antifungal activity against Saccharomyces cerevisiae and C. albicans, and also identify these bioactive metabolites and elucidate their structure. I'll also determine the mechanism of action of these metabolites and determine their cytotoxicity to human cells, and then identify the fungal species producing this metabolite. So, for the methodology, seaweeds were collected from 4 beaches in Ghana. That's Adan, Keta, Anomabu, and Pampram. And these weeds were sterilized, grinded, and plated on agar. So pure isolates from these agar plates were put in broth cultures, and after 4 weeks, they were extracted with ethyl acetate. The crude extracts were then tested against C. albicans and S. cerevisiae using the Dix diffusion method. Some of the pure isolates were also grown on large cultures so that we can use them for product isolation. That's identification through mass spectrometry and then structural elucidation using NMR. We will also be determining the mechanism of action of the metabolites using a panel of mutant yeast and also a panel of chemical phenotype modifiers, and also determine the cytotoxicity of the metabolite. So far we have isolated 121 MEF strains and tested these 121 strains against C. albicans and S. cerevisiae. and we came out with a plot in order to select the best performing isolates. So this is a plot of size of zone of inhibition against the number of extracts producing these zones. So if we even put the cutoff points as extracts producing zones greater than 15 millimeters, we have 41 of them that we are working with right now. So we are still working on this 41 in order to select the best. We tested it on SRVC, all the 121 isolates on SRVC, and the good news is that the isolates were more active against the pathogenic fungi, which is C. albicans, as compared to the SRVC, which is the non-pathogenic fungi. So research has shown that certain compounds are able to help bacteria become more sensitive to the antibiotics. So we wanted to test the same thing using standard antifungal compounds. So we have a selection of compounds that we modify media with in order to test this same thing. And we realized that artemisinin and then vitamin C all cause an increase, cause the C. albicans to be more sensitive to the standard antifungal compound, fluconazole. We would be replacing the standard antifungal compounds with our isolated compounds very soon. And this will also help us to understand the mechanism of action of our compound. We tested the same thing using SRVC as well, and for SRVC, it was artesunate and then artemisinin that caused the SRVC to be more sensitive to the standard antifungal agent amphotericin B. So we went ahead and then selected a few extracts from our round 3 collections, and then we tested them against mutant C. albicans strains. And what we have seen is that the mutants were more sensitive to the extract as compared to the wild type. We will eventually be replacing the extracts with our compounds when we find them to get a better understanding of what the compounds are targeting in the organism. So we did a bit of fractionation, and so far we have 6 active fractions against C. albicans, and then 4 active fractions against S. cerevisiae. So in summary, we have isolated 121 marine endophytic fungi, and these are very active against C. albicans and S. cerevisiae. And we have 41 of the 121 that produce zone greater than 15 millimeters, which we are still working on. And we have now seen that some chemical phenotype modifiers increase the sensitivity of yeast to standard antifungal agents. We have 8 active fractions out of the 42 fractions from 6 organisms. And so my study will lead to the discovery of new compounds which can be used as drug for the treatment of fungal infections. And then the work will provide a better understanding of the mechanism of action of these compounds. And then new mechanistic activities for existing chemical phenotype modifiers will be determined. I would like to acknowledge my supervisors, Dr. Patrick Atta, Dr. Ajimeni, Dr. Dokas Osei-Safo, Prof. James Ajay, WAGBIB, and also Biochem Department. Thank you.
Thank you very much, Etta, and also being on time. Yes, we have time to look at 2 or 3 questions. Any comments? Any questions? Any questions?
I think I missed this. The 6 compounds that are active against C. albicans and yet 4 against S. cerevisiae. Mm-hmm. Do they overlap?
Yeah, they do. They do overlap.
And you have plans to see if they're toxic in general against?
Yeah, human cells.
Any?
Thank you. Following up on our discussion earlier, actually,
yes.
Do you have any idea on the concentration of what's in there? Your compounds, the concentrations?
Yeah, I do.
Would you like to share?
Yeah. Okay, I can't. Oh, it's not. I can't. I have a table of all of them because I take a specific amount for testing, like 30 microliters. So I know the weight of the crude extract, so I'll be able to project the mass concentration from there. Okay.
Yeah, the bulk fermentation.
Yes, sir.
To what extent would that affect the metabolites or chemicals compared to maybe the smaller vessels?
Yeah. So we've so far seen that for the MEFs, if you culture them in bulk like that, the metabolite activity reduces. So that's what we have observed in just like a few months ago. So we from now on we would think that we have to culture them in very, very small quantities.
The condition I realize that this way seals.
No, we usually cap them with perforated caps and then use Wattman filter paper to cover it so that there will be gaseous exchange by all means in them.
Yes, thank you very much, Etera, for the presentation. So we move on to the last of the four fellows. The next one is going to be presented by Dr. Adjoa Asantipoku. She will present on molecular epidemiology of Mycobacterium africanum in Ghana.
Good afternoon. So I'm the last of 4 strong women presenting for this section, and I hope I'll make the women proud. Yes. Okay, so I'm here to present on molecular epidemiology of Mycobacterium africanum in Ghana. First of all, I will tell you the global burden of TB. TB is a disease that is found in all continents. When you look at the map that I've projected right now, you see that the greatest burden is in Africa and in Asia. And together, these 2 continents contribute about 80% of all TB cases recorded annually. In 2014, TB recorded 9.6 million cases, of which 1.5 million deaths occurred. Now with TB, we have 2 million latently infected people walking around freely. So if stringent measures are not put in place, these group of people would provide a vast container for active transmission of TB. You may ask what causes TB. Before that, the same can be said for Ghana. TB is still a public health disease in Ghana. Recently, after a prevalence study, we now know that Ghana, we have a prevalence of 282 per 100,000 population, and a TB HIV prevalence of 24%. This actually makes Ghana one of the highest TB and TB HIV high burden countries in sub-Saharan Africa. Now to the next slide. You may ask what causes TB. TB is caused by a group of closely related bacteria, collectively known as Mycobacterium tuberculosis complex. For us in WACBIB and in Noguchi, we are interested in those that cause TB in humans, not the ones that cause TB in animals. So for the humans, we have 7 main lineages causing TB in the humans. And when you plot these particular lineages as are projected here against geographic regions, you'll find that there are some that are more distributed as against some that are restricted to certain sites. If you look closely at the map, you can see that the red dot, which represents lineage 4, which is the most dominant lineage, it dominates the whole world, as against the blue dot, the pink, the purple, the green, and the brown. For us in West Africa, we are mostly interested, in addition to the red dots, to the blue and the brown dots. These 2 dots represent 2 lineages of the human causing TB, collectively known as M. africanum. M. africanum is found only in West Africa. It was discovered in 1968 in Senegal, and initially it was described as an intermediary between M. tuberculosis and M. bovis based on some shared characteristics. But in this current era of genomics, we have now seen that it is actually very distinct from these two particular pathogens, as distinct as the human-causing pathogen is from the animal-causing TB pathogen. So in this genomic era, what else have we found? We have found that compared to the M. tuberculosis, which is more dominated globally, M. africanum has an ESA-cis attenuated response. ESA-cis is the early secretory antigen, and M. africanum has an attenuated response to it. It progresses slowly from disease to from infection to a disease. And for a carbon source in an artificial media, M. africanum prefers pyruvate as against glycerol. Currently, a PhD student in our group also discovered that there are some differences in metabolic genes between M. africanum and M. tuberculosis. These differences could probably explain why there's a slower progression from an infection to a disease when you're infected with M. africanum. Although we say M. africanum is restricted to West Africa, its distribution in the West African countries is not the same. If you look at the map that I've projected here, you can see that towards the western part of West Africa, we have one particular lineage of M. africanum known as the lineage 6. That is the green dot that I showed earlier on. And to the eastern part of West Africa, you have basically M. africanum 5. That is the brown dot that I projected earlier on. But there are few countries in between, that is Ghana, Benin, Ivory Coast, that have both M. africanum 1 and M. africanum 2, or the lineage 5 and the lineage 6 in abundance. Although scientists have been able to come up with all these differences and all these research findings, one particular finding has eluded scientists up till now, and that is why M. africanum is restricted to West Africa, why it never left West Africa with our brothers and sisters who were shipped out to the Americas and to the European countries. All the other lineages were shipped out with our brothers and sisters, but M. africanum somehow remained in West Africa. Could it be that it found a particular niche in West Africa? Or could it also be that it was out-competed by the other lineages when it got to the Americas and to the Europeans? Up till now, we haven't been able to come up with a particular answer. Now, with Ghana harboring both variants of the M. africanum in substantial amounts and also harboring all the other lineages, this gave us an opportunity to study this particular question and probably come up with a plausible answer. So what did we do? We collected smear-positive, sputum from smear-positive TB patients that report to the clinics here in the southern part of Ghana. So we took samples from the central region, from the western region, from the greater Accra region. In addition, we also collected biological and epidemiological data from the patient after informed consent. The sputum that we obtained were cultivated. We did lineage identification by molecular assay, and then we analyzed all the data that we got by multivariant analysis. For the molecular techniques that we used SNP typing for the main species identification, and then we used polygotyping for the subspecies identification. So polygotyping is one assay that is unique to Mycobacterial tuberculosis complex. It is based on the presence or absence of specific 43 spacers in the direct region. So what did we get? Well, we found that, yes, Ghana has all the 6 lineages here in our country, and then we also found that M. africanum, from our population, M. africanum causes about 17% of all the TB cases. This is in contrast to reports from neighboring countries where they see a very sharp decline of M. africanum from the 1960s to the current 2014, especially in Cameroon. Initially, Cameroon was reporting the highest prevalence of M. africanum it was reporting around 48%. But currently, Cameroon is reporting 9% of all TB cases to be caused by M. africanum. You could ask why. Maybe it could be that initially the analysis was being done using biological assays. But right now, with the genomic era, using genomic assays, they found out that actually M. africanum was causing less. But in Ghana, what we got confirms what was reported in 2011 by Yeboah Menu, where she says that M. africanum causes about 18% of all TB cases in Ghana, and also confirms what Addo et al. Also reported in 2010, which says that M. africanum was causing about 22% of all TB cases in Ghana. So it hasn't dropped that much. We are still within the 20% margin. If you look at the epidemiological features of M. africanum, you can see that for the first time we found an association with an ethnic group and M. africanum. And here I want to make a disclaimer. I'm not saying Evers are more prone to getting TB. What we are saying is that from our data set, we saw that there was an association between a particular ethnic group and TB caused by M. africanum. One could also ask that just looking at this small data set, which was 622 patients, could it be that just one short study, this association could be by chance? Just because you did it once and you saw this association doesn't mean that it's a biological association. So we looked at our spoligotyping patterns, and we saw that with this particular association, we have different patterns. So it could not be that it's by chance, and it could not also be that it's a single outbreak. And when we plotted the different lineages that we got against the residential addresses of the patients, we saw an even distribution of the M. africanum from the Greater Accra to the Western region. So it could be that there was something really going on that we don't really know. So we repeated this study using a larger sample size of 1,200, more than double what we used, and we still saw the same association. In this particular study, we saw M. africanum causing about 22%, but we still saw the association between the Evers and the Evers being more susceptible to TB caused by M. africanum. So then we said that, well, it could be that this particular association is more biological than just by chance. In addition, we also saw an association between lineage 6 and HIV. The current prevalence rate of HIV in Ghana is 1.9%, but if you look at TB, it's 24%. So, and we also saw that lineage 6, over the years, the prevalence of lineage 6 in Ghana has been on the rise. It could be that this particular association with HIV is pulling up the lineage 6, but when you get HIV, you are immunocompromised. And with TB, the most virulent one is the one that progresses from infection to a disease. So if you're immunocompromised, then it's an even playing field for all the lineages. Anyone can come up as compared to somebody who is immunocompetent. So the significance of my study, we find out that M. africanum is still present in Ghana at almost the same prevalence of 20%. Within the regions that we worked, it was uniformly distributed. And for the first time, we reported an association between ethnicity and M. africanum. Could this be an indication of host pathogens or host factors playing a role here? We also confirmed what was said in Gambia, where we say that TB patients infected with lineage 6 were more likely to be HIV positive. And although I didn't present this data here, we also found M. africanum. We also extracted M. africanum from animals. This confirms the public health importance of M. africanum. So now this feeds into my postdoc study, where we want to establish a host genetics TB study in Ghana to identify candidate genes or genetic polymorphism that are unique to M. africanum. This will give us an idea why M. africanum is restricted to West Africa. And in this era where vaccines are being developed, we think this is very, very important so that they will not develop a vaccine that might not work in Ghana or in West Africa, where you have M. africanum restricted, but not M. africanum cannot be found in the other parts of the world. I would like to acknowledge Professor Yeboah Menu's group for taking me through all this work. And then Professor Ambrose Wankam for agreeing to be my mentor for my postdoc. Most of the work will be done in his lab. And without all these collaborators and all these founders, I would not have been able to do that work. And I thank WorkBig for agreeing to pay for my postdoc studies.
Thank you very
And
much.
the last slide is the slide.
That was a very interesting presentation. We have time. We are behind time. We have time for one or two questions.
Madam, thank you for the question. I have an issue with having a lab-based research linked to a particular socio-demographic characteristics like ethnicity. Because I'm not very sure whether your study is adjusted for occupation and other issues. Because there could be correlation between the particular ethnic group and particular occupation which they are doing, which probably posing them at risk to such an infection. So I guess it's a bit problematic, especially when it's not a large-scale epidemiological study. So probably you may want to hide the identity for a while and then do better and larger scale study to be able to establish, especially in this country where a lot of things are politically linked and you have huge ethnic. I guess it's the earlier that so that you can see the ethnicity, but you don't necessarily need to mention the ethnic group.
Okay.
So I guess you need to have a look at that and see how we present such data in the near future. Thank you.
Thank you very much.
Thank you. So, very interesting study. The second study, you say you used 1,500 samples.
200.
1,200 samples. Do you have idea of the geographical
Yes.
distribution
Yes.
compared to the first study?
Yes, so the second study, we took the samples from Greater Accra and from the Northern region.
Okay. So, thank you very much, Adwa. So that ends the session. Our last presentation will be given by Lashvid. Lashvid is a professor of immunology and parasitology at the Center for Medical Parasitology, University of Copenhagen in Denmark. Lash has had a very long collaboration with University of Ghana. She's been part of the research work here at Noguchi since 1991 and 1992. He helped establish the research unit at Kolebu and also site at Dodowa and also Hohoi of late. Lash.
Thank you very much. First of all, I would like to emphasize that I'm extremely proud and happy to be here at this conference. Ghana is my favorite African destination. I have been coming here regularly for the last 20 years. And I'm happy to be given the opportunity to talk on my favorite subject, namely immunity to malaria and the value of collaboration. We heard yesterday that Keith Gall emphasized the need for capacity building, and I will strongly agree with him on this. And we have also heard from Solomon Ofori-Akwa and from Ambrose Wankam the need to do research on African diseases by Africans in Africa. And I was very happy that Ambrose said that we are, in fact, all Africans. So I do not need to feel out of place here, even though, obviously, I have been out of the sun for a while. Anyway, I will get right into it. I will talk to you a little bit about the falciparum life cycle, not that you don't know it, but why it could be interesting in terms of protective immunity, in terms of both naturally acquired protective immunity, and also in terms of vaccine-induced immunity to malaria. And then I will talk a little bit, not so much on the value of North-South collaboration, but the fact that we are at this conference today, I think, is evidence of the value of North-South collaboration, because you could say that our main commander at this meeting, Gordon, is definitely a product of a very fruitful North-South collaboration from his PhD studies in the United States. Gordon yesterday told you that I've known him for a very long time, which is true, and that I considered him a joker initially, which is not true. I think I have never really been in doubt that Gordon would go far, and he has gone very far. But he is living proof that you do not have to be dull to be a scientist. And I hope you will agree with me on that. This is a very classic slide on the development of immunity to malaria, and you will see it in most talks, and we have seen it briefly already. I will not really dwell on it very much, but I think there are some features that are important here. That is that there is an initial period for the first 3 to 6 months of life where infants are really quite resistant to malaria. This is followed by acquisition of protective immunity, but that is really excruciatingly slow. It takes years to develop, and it never really becomes sterile or solid. There are many people who believe that the protection that you acquire is short-lived. Whether that is really true, I don't know. It depends a little bit on how you define protection, I think. Peculiarly, it seems to be easier to acquire immunity to severe disease than to uncomplicated disease and asymptomatic parasitemia. The acquisition, as was alluded to yesterday, also very much depends on the endemicity, and also the pattern of disease severity also varies with endemicity. So clearly, the way that you become immune to malaria is in many ways quite different from your textbook examples of how you acquire immunity to infectious diseases. I think that much of this knowledge derives from studies in Africa, and the way that we can get this sorted will come from studies in Africa. It's been known for a very long time that antibodies play a key role in the acquisition of immunity to malaria, going back to the studies done by Ian McGregor and Sidney Cohen back in the 60s, where they injected children with malaria with immunoglobulin from clinically immune adults and showed that that could really reduce parasitemia quite dramatically. These experiments also showed that it is actually the asexual blood stages that are the main targets of this immunity. So let's look a little bit at the life cycle in terms of what are the potential targets of immunity of malaria. The infection starts, as you know, when an infected mosquito injects sporozoites. The sporozoites could certainly be a target of antibodies. They are covered in a protein that has been known for a very long time. They then move on to infect the liver. Antibodies might play a role there, but probably, if anything, it's more cell-mediated immunity. And then there is, of course, the merozoites that we have already heard a lot about, and the intracellular, the intraerythrocytic stages of the parasites, which we've heard less about until now. The gametocytes, which are the stages that are transmitted back into the mosquito, could also be targeted by antibodies, theoretically, and so could, in fact, also the stages that are present in the mosquito. However, if you look at the way that naturally acquired that immunity to malaria is acquired after a natural exposure to the disease, all the immune responses, by and large, are concentrated on the asexual blood stages, as you might wonder why that is. And I think that the reason for this is that this is what the immune system considers to be the dangerous part of the life cycle. This is the stages that are associated with all the clinical symptoms of the disease. So let's look a little bit at the asexual multiplication cycle. In falciparum, it's a 48-hour cycle. I don't need to go into the details here, but let's just start with the merozoites that have already been touched upon a little bit. And you cannot see this, so I will read this. There are the potential that there could be invasion-blocking antibodies that would prevent the merozoites from getting from one erythrocyte into the next. There could be opsonization by antibody or by complement that could allow the phagocytic system to destroy these, and there could be direct complement lysis. So the